Hello everyone, and thank you for inviting me to give this talk about our Greenlandic breast cancer study. Before I start my presentation, I would like to thank everyone who has been a part of the study. Without their valuable contribution, the study would not have been possible. Breast cancer incidence in Greenland has generally been lower than in other Nordic countries. However, during the last years, there has been a steep increase in the incidence, much higher than in the Nordic countries in general. During the last 10 years, the annual change has been around 6.5% compared to small decreases in both Denmark and the Nordic countries in general. Breast cancer is a multifactorial disease with many established risk factors, which I will not go into detail with here. However, the established risk factors only explain a fraction of all breast cancer cases, and twin study has suggested that environmental factors may explain a large part of breast cancer cases. In our study, we focused on the effect of contaminants, genetic factors, and diet. I'll just give you a short introduction to Greenland, where we conducted our study. Greenland has a population of 56,000 people, which live in settlements and towns around the coast. 1/3 of the population is living in the capital, Nuuk. Greenland has experienced a rapid cultural and social transition from a hunting and fishing society to a society highly influenced by the Western world today. It has resulted in a decrease in the traditional Greenlandic food items and a more sedentary lifestyle. There's also been a change in disease pattern with an increase in lifestyle-related diseases such as diabetes, and also an increase in lifestyle-related cancers such as lung cancer, colon cancer, and breast cancer. The traditional Greenlandic diet has been dominated by marine mammals and fish, which contain several both pro-carcinogenic and anti-carcinogenic components. The marine mammals has high level of omega-3 fatty acids, minerals, and vitamins, which has been associated with reduced breast cancer risk. But it also contains high level of persistent organic pollutants and heavy metals, which may increase the breast cancer risk. The imported food items mainly comes from Denmark and consist of sugary foods and drinks, milk, meat, fruit, and vegetables. The persistent organic pollutants is a large and heterogeneous group of chemicals which are slowly degraded and bioaccumulate in human and animals. We evaluated the effect of 3 groups: the PCBs, the organochlorine pesticides, and the perfluorinated alkylated acids. The first 2 groups are lipophilic and accumulate in adipose tissue, whereas the PFFAs are amphiphilic and accumulate in organs such as the kidney and liver. The main exposure is food, and in Greenland, marine mammals are the major food exposure source. Greenland has some of the highest levels, both compared to the world in general, but also when comparing with other Arctic regions. Several of the PERP has been classified as carcinogenic, probably carcinogenic, or possibly carcinogenic by IAC. We used a case control study design and included participants of Greenlandic Inuit descent. We recruited in 2 periods, from 2000 to 2003 and from 2011 to 2014. We included nearly 100 controls and 100 cases, which correspond to around 80% of all breast cancer patients in Greenland during this period. The controls were either healthy women or non-cancer patients from the hospital, which were matched on age and residential area. We had participants from all parts of Greenland and had both biological samples and questionnaires. Now to some of the results. I'll start with the influence of PUP exposure. We found a higher level of PUPs in cases compared to controls. This was shown for all the three chemical groups, the PCBs, the organochlorine pesticides, and the perfluorinated alkylated acids. Similarly, we found increased breast cancer risk with high level, same level of PUPs. In the third total, in breast cancer, risk was significantly higher than in the first total. For the organochlorine pesticides, we saw a similar trend. However, it was not statistically significant, only in the analysis with continuous variables. We then measured the effect of PUP mixtures on hormone receptor activities and assessed the association with breast cancer risk, as we would like to elucidate if the PUP-induced breast cancer risk were through their endocrine disrupting effects. We did this by extracting the actual mixture of PUPs from the blood samples of cases and controls in fractions and determined the estrogenic, androgenic, and aryl hydrocarbon receptor activities of the mixtures. We found that the estrogen activity were not related to breast cancer risk, but the androgen receptor activity seems to be related. We found that the percentage of samples with decreasing androgen activity were higher for the controls than for the breast cancer cases. And the group with decreasing androgenic activity had a reduced breast cancer risk. We did also see that the dioxin-like activity were related to breast cancer risk. The cases had a lower dioxin-like activity than the controls, and the third total had a decreased, reduced breast cancer risk compared to the first total. Now to the study of genetic variations. We selected variations in phase 1 and phase 2 enzymes together with the BRCA1 Greenlandic founder mutation and associated the effect on breast cancer risk. The evaluated variations affected the activity of the enzymes and thus the hormone level and level of reactive compounds in the body. The sub-17A1 and sub-19A1 increased the enzyme activity with the variant allele, meaning that the estradiol and testosterone levels were higher in variant carriers. The sub-1A1 and 1B1 had also a higher activity in the variant allele, resulting in a lower hormone levels, but a higher level of reactive compounds. And the COMP variant allele had a lower enzyme activity, resulting in a higher level of reactive compounds in the body. We found that the BRCA1 Greenlandic founder mutation were related to breast cancer risk. The heterozygotic variant carriers had a higher breast cancer risk of 12.2 compared to the homozygotic non-carriers. The sub-17A1 were also related to breast cancer risk. The variant allele carriers had a reduced breast cancer risk compared to the homozygotic non-carriers. None of the other 4 variants were related to breast cancer risk. We then analyzed the modifying effect of the variant alleles on the association between POPs and breast cancer. We did find tendencies of a stronger association in some genotype strata. The tendencies were most pronounced for the perfluorinated alkylated acids, where we found that the TT stratum of sub-17A1 had a higher risk compared to the variant allele carriers. And similarly, for the sub-1B1, the variant allele carriers had a higher risk, a stronger association with PFAS than the homozygotic non-carriers. Now we evaluated the DNA methylation patterns and the relationship to breast cancer risk, POP levels, and other breast cancer-related factors. We selected several genes based on literature search, the repetitive elements, breast cancer-related tumor suppressor genes, circadian genes, and the estrogen receptors. Most of the evaluated genes were not related to breast cancer risk. However, the methylation of the repetitive elements were related to breast cancer. For line 1, we found a reduced breast cancer risk in the 3rd tertile compared to the 1st tertile. And also for allo, we found similar tendencies which were non-significant. The POP levels were related to the DNA methylation. We found generally positive associations. However, for one of the groups of the perfluorinated alkylated acids, the carboxylated acids, we found both negative and positive associations. We hypothesized that this association may indicate that POP exposure is influencing breast cancer risk through the changes in DNA methylation. And now to some of the conclusions. We found that several factors influence breast cancer risk to some degree in Greenland. Both the POP level, which were positively associated with breast cancer risk, the receptor activity of the actual POP mixtures in the body, the genetic variations of BRCA1 and phase 1 enzymes, and also the global DNA methylation were related to breast cancer risk. Of note, I want you to notice some strengths and weaknesses in our study. We found our breast cancer cases were confirmed with histological reports. We only included Greenlandic Inuit women, and we had good and reliable exposure measurements with biological samples. However, we used a case control design. We included controls which were both healthy women and non-cancer patients from the hospitals. And of course, due to the small population in Greenland, we had a small sample size with low statistical power in some analyses. At the end, I want to say Røynak, thank you, and you're very welcome to contact me on email if you have any comments or questions to our study. Thank you.